Assalamu alaikum. Today I will give you a lecture about the embryology of skeletal system. The skeletal system developed from the paraaxial and lateral plate mesoderm and from neural crest. Paraaxial mesoderm then form the somatomere in the head region and somite from the occipital region caudally. This is the somite. The somite differentiate into ventromedial part, which called sclerotum, and dorsolateral part, the dermomyotome. This is the sclerotum, and this is the dermomyotome. This is the ventral, and this is the dorsal part. Then, the sclerotum become polymorphous and form a loosely woven tissue. They call it mesenchyme or embryonic connective tissue. These then differentiate into fibroblast or chondroblast or osteoblast. This is the sclerotum, as we mentioned, which differentiate into mesenchymal tissue. In a flat bone of the skull and the ribs, the mesenchyme differentiate directly into the bone. So the mesenchyme differentiate and forming the bone directly, which called intramembranous ossification. In other bone, which I mean other long bone, the mesenchyme first give rise to hyaline cartilage, which then ossified by endochondral ossification. So we have two methods of ossification. We have intramembranous ossification, and this is intramembranous, and we have endochondral ossification. Intramembranous ossification occur in flat bone of the skull and the ribs, while endochondral ossification occur in a long bone. Skull divided into neurocranium, which form the protective case around the brain, and viscerocranium, which form the skeleton of the face. In a newborn, at birth, the flat bone of a skull are separated from each other by a narrow seams of connective tissue, which called sutures. At point where more than two bones meet, the sutures are wide and called fontanelles. We have anterior and posterior fontanelles. These are the sutures and these are the fontanelles. This is anterior fontanelle, this is posterior fontanelle. These are the sutures. These sutures and fontanelles allow the bone of a skull to overlap during birth. This method called molding. Development of the limbs. At fourth week of development, the limb bud become visible as outpocket from the ventrolateral body wall. It consists of mesenchymal core, which will form the bone and connective tissue of the limb, and it's covered by a layer of cuboidal ectoderm. This ectoderm, at the distal border of the limb, thickened and formed the apical ectodermal ridge. This is the limb bud and the ventrolateral part. This is the limb. This apical ectodermal ridge exerts an inductive influence on the adjacent mesenchyme, causing it to remain as a population of undifferentiated, rapidly proliferative cells, which called the progress zone. At 6 weeks, the terminal part of the limb bud become flattened to form the hand and foot plate, and it's separated from proximal segment by circular constriction, which form the wrist joint. This is the limb bud. This is the apical ectodermal ridge, and this is the hand and foot plate, and this is the constriction will form the wrist joint. Then a second constriction divide the proximal portion into two segments, and then the main part of the limb can be recognized. The fingers and the toe are formed when a cell death in the apical ectodermal ridge separate this ridge into a 5-part. Development, this is important, هاي شيء مهم. Development of upper and lower limb is similar, except the formation of the lower limb is 1 to 2 weeks behind the upper limb. I mean, the lower limb is formed later than the upper limb. During the 7th week of gestation, the limb rotates in opposite direction. So the upper limb rotates 90 degrees laterally, so that the extensor muscle lie on the lateral and the posterior surface, and the thumb lie laterally, while the lower limb rotates 90 degrees medially, which places the extensor muscle anteriorly and the thumb medially. At 6 weeks of development, the bones of the extremities are formed from the chondrocyte. The ossification of the bone, of the long bone, endochondral ossification, begin by the end of embryonic period. The primary ossific center. We have a primary and we have secondary ossific nucleus or ossific center. The primary ossific center are present in all long bone by 12 weeks of development. The primary ossific center are present in all long bone of the limb by 12 weeks of development. From the primary center in the shaft or diaphysis of the long bone, the chondral ossification gradually progresses toward the end of the cartilaginous model. At birth, the diaphysis of a long bone is completely ossified, but the two end, the epiphysis is still cartilaginous. Then, ossific center or ossific nucleus arise in the epiphysis, but temporarily a cartilage plate remains between the diaphysis, which already ossified, and the epiphysis, which call this plate, they call it epiphyseal plate. This plate is important for growth in the length of the bone. The endochondral ossification proceed on both sides of this plate. When the bone acquired its full length, the epiphyseal plate disappears, and the epiphysis unite with the diaphysis. Regarding the development of the vertebra and vertebral column, the vertebrae form from the sclerotom portion of the somite, which are derived from the para-axial mesoderm. At 4th week, the sclerotom cells migrate around the spinal cord and notochord to merge with the cell from the opposing somite in the other side of the neural tube. This is the sclerotom portion. So the neural or vertebra form from the sclerotom portion also. Then the sclerotom portion of each somite also undergo a process. They call it resegmentation. It's a process when the caudal half of each sclerotom grow in two and fuse with the cephalic half of each adjacent sclerotom. So each vertebra is formed from the upper and lower sclerotom. So the lower or the caudal half of upper sclerotum unite with the cranial half of lower sclerotum to form the vertebra. Thus, each vertebra is formed from combination of caudal half of one somite and the cranial half of the adjacent one. Mesenchymal cell between cephalic and caudal part of original sclerotum segment does not proliferate, but fill the space between precartilaginous vertebral body and they form the intervertebral disc. This is how the intervertebral disc develop. Although the notochord regress in the region of vertebral body, but it persists and enlarge in the region of intervertebral disc and forming the nucleus pulposus, which is then surrounded by circular fiber of annulus fibrosus and the combination of these two structures forming the intervertebral disc. Regarding the muscular system developed from the mesodermal germ layer. It consists from skeletal, smooth, and cardiac muscle. The skeletal muscle derived from paraaxial mesoderm, which forms somite from the occipital to the sacral region. This is the somite. And this is the sclerotum. The smooth muscle differentiated from the subplanchnic mesoderm that surround the gut. Cardiac muscle is derived from the subplanchnic mesoderm that surround the heart tube. Striated skeletal muscle, which are the muscle that form in the upper and the lower or the musculoskeletal system. The somite and somitomere form the muscular of the axial skeleton, body wall, and the limb and the head. From the occipital region caudal, the somite form and differentiate into the sclerotum and dermomyotum and two muscle forming region. So from occipital region caudally, the somite form and differentiate into sclerotum and dermomyotum and the two muscle forming region. So the somite differentiate into sclerotum and dermomyotum and two muscle forming region. One of these muscle forming region appear at the ventrolateral edge, or they call it ventrolateral lip. And the cell from that lip contribute to formation of the myotome and also provide progenitor cell for limbs and body wall musculature. The other region lie at the post-dorsomedial lip. They call it dorsomedial lip. And these cells migrate ventral and contribute to formation of myotome, and eventually these cells will form the muscle of the back. So this is the myotome and this is the dermomyotome, and this is the muscle bud, including the myotome and the dermatomes also. By the end of 5th week, the muscle cells are collected into two parts. Small dorsal portion, they call it epimere, which form from the dorsomedial cell of the somite that recognize as myotome. This is the myotome. Large ventral part, they call it hypomere, which formed by migration of dorsolateral cell of the somites. So this is the epimere, and this all is the hypomere. The epimere will form the muscle of the back of the spine. The nerve innervating segmental muscle are also divided into the dorsal primary ramus for the epimere or the muscle of the back. Ventral primary ramus for the hypomere, which form the muscle of the limbs and the body. The myoblast of epimere form the extensor muscle of the vertebral column, and those of the hypomere give rise to the muscle of the limb and the body wall. So, this is the epimere, which form the muscle of the back and supplied by the posterior ramus or the dorsal ramus, and this is the hypomere, which form the muscle of the limbs. This is the limbs, and also form the muscle of the body wall. Limb musculature. The first indication of limbs musculature is observed in the 7th week as condensation of the mesenchyme near the base of the limb bud. The mesenchyme is derived from the dorsolateral cell of the somite that migrate into the limb bud to form the muscle. With elongation of the limb bud, the muscle tissue split into a flexor and extensor component. The upper limb bud lie opposite the lower 5 cervical and the upper thoracic segment. The lower limb bud, the limb bud of the lower limb, lie opposite the lower 4 lumbar and the upper 2 sacral region. So this is the upper limb and this is the lower limb. So the upper limb lie opposite the cervical, while the lower limb located opposite the lumbar and sacral region. As soon as the bud form, the ventral primary rami from the appropriate spinal nerve penetrate into the mesenchyme. At first, each ventral ramus enter with isolated dorsal and ventral branch, but soon these branch unite to form a large dorsal and ventral nerves. Thus, the radian nerve, which supply the extensor muscle, is formed by combination of a dorsal segment branches, while the ulnar and median, which supply the flexor muscle, are formed by a combination of ventral branches. The spinal nerves not only play an important role in differentiation and motor innervation of the limb musculature, but also provide sensory innervation for the dermatomes. This is how the upper limb located opposite the cervical and the lower limb located opposite the lumbar and sacral. And thank you very much.